All right, so today we're going to be looking at one group of how to classify called cladistics and clades and different ways that scientists use to figure out some classification ideas. So some characters used for classification are in this chart here. So morphology are the different shapes and structures. So looking at the shape of a plant seed. Anatomy, cytology is looking at cells. Phytochemistry are special organic compounds that maybe only plants have. Chromosome number, and molecular differences, which is going to be a key one. So molecular systematics is classifying organisms using molecular differences. So looking at things like their protein and their DNA and how the strands are different. So we'll get more into that molecular part in just a bit. So cladistics is a system of classification that groups taxa together according to the characteristics that have evolved most recently. So again, the main part is looking at how they evolved. So this is natural classification that we brought up before. So common descent is crucial for deciding into which groups to classify organisms. So what did they descend from? What are their common ancestors? So to decide how close a common ancestor is, researchers look at how many primitive and derived traits that an organism share. So a primitive trait is a plasmomorphic trait. It's characteristics that have the same structure and function and that evolved early on in the history of the organism being studied. So a good example of this in plants are leaves with vascular tissues, the xylem and phloem, that evolved very early on in plants to transport liquids, water around the plant. So that's a primitive trait. Okay, they have the same structure and function in all plants. A derived trait or apomorphic trait are characteristics that have the same structure and function, but that have evolved more recently as modification of previous trait. So they've been derived. So flowers, for example, evolved much later than leaves. So they evolved more recently than the leaves with vascular tissues, and scientists actually think they're adaptations of vascular leaves. So the leaves kind of adapted and changed into flowers. Okay, so primitive came earlier, derived traits came later. By comparing such characteristics, quantitative results show which organisms have a more recent split in the evolutionary past and which have a more distant split. So when did they split evolutionarily? So a clade is a monophilic group. So mono meaning one, okay, group composed of the most recent common ancestor of the group and all of its descendants. So it's one way we can group them together based on a common ancestor. So when a group can be split into two parts, one having certain derived traits that the other does not have, the group forms two separate clades. So as soon as we get some sort of derived trait that's different, we're going to separate them into two groups or two clades. Clade can have just one species, or often it can be made up of multiple species, just depending on the common ancestor. So biochemical evidence of clades. So DNA is very important. Every known living organism on Earth uses DNA as its main source of genetic information. So this is compelling evidence that all life on Earth has a common ancestor that use the same basic pattern of DNA. So proteins, all organisms use the same 20 amino acids to form their proteins. Amino acids can have two orientations, a left-hand or a right-handed orientation, as shown here, kind of the direction that they're sitting with the hydrogens on opposite sides. Majority of organisms use the left-handed amino acids. Some bacteria use right-handed, but we can see most are going to use one type. Again, that suggests a common ancestor. Chemical similarities implies a common ancestry for all life forms that use the left-handed amino acids. Okay. So phenology is the study of evolutionary past of a species. So species that are most similar are most likely to be closely related. Comparing the similarities in the polypeptide sequence of certain proteins in different groups of animals to trace their common ancestry. So for example, looking at proteins like hemoglobin, cytochrome C, chlorophyll, those are all very common proteins, so we can actually use those to trace similarities. Biochemical phenology is the study of nucleic acid sequences in DNA and mitochondrial DNA. Remember, there is DNA in the mitochondria, so that can be traced as well. So changes in the DNA sequence of genes from one generation to the next is partly due to mutation. And the more differences there are between two species, the less closely related the species are. So the more differences, that means there's more mutations, so more time has evolved between the two, making them more different.
A phylogenetic tree is a visual representation of the relatedness of species by descent from a common ancestor. The branch tips represent existing species. Branches connect a common ancestor, and the nodes depict where one species differentiated two or more species. The arms not guided by knowledge.
Okay. So I know that was a little bit long, but it does go through a lot of things we've talked about, and we'll talk about the cladograms in our next lesson and how to make those. All right, so this is a good example from your textbook that shows comparing DNA sequences for 4 different organisms. So here we have species 1, 2, 3, and 4, and the red is showing the mutation or a change. So notice 1 and 2 are very similar. They only have one difference here with the A as one mutation. So they are more closely related, but at some point there was a split. So 1 does not have that change to the A, and the rest do. So that's why we have this split here. But 1 and 2 are more closely related. And then eventually we get species 3 has that change of A, but it also has the G and C change mutation, and species 4 has several more mutations. So we can see how we made this tree this way. Okay, so species 1 and 4 are the most different because there are more mutations between 1 and 4. So that's just a way to show the differences, mutations, and how we're representing those 4 different species. All right, so evolutionary clock, so differences in polypeptide sequences, which are going to be in proteins, accumulate steadily and gradually over time as mutations occur from generation to generation, as we saw in that last page. So changes can be used as kind of a clock to estimate how far back in time 2 related species split from a common ancestor. So we can compare homologous molecules from 2 related species, so similar, and count the number of places along the molecule that they are different, as we kind of just looked at with the DNA sequence before. You can also look at mitochondrial DNA. It's good to study because it is not shuffled and mixed during meiosis or fertilization. It's passed directly from the mother to child without modification. So any changes are only due to mutations. So it's a good place to look for mutations because we're not having other mixing of DNA because of fertilization. DNA hybridization is a technique used to measure the differences in DNA. So to do this, they can take one strand of DNA from species A and a similar strand, homologous strand from species B, and fuse them together. When they do this, base pairs connect. There's a match, so we know that they're same. So A and T pair up, C and G pair up. But there are places where they're going to repel and don't connect, and this is where there's a difference in the DNA sequence and there is not a match. So very quickly, they can see where the places where they're different. So here's a good example of that. So DNA from species A, species B, you can see areas where they're matching, we get the green check mark, and areas where they're repelling that they don't match. So we can count how many places they're not matching. Okay, and then here they did a little bit more. We have different sequences here. And just to kind of show the split, the idea of using quantitative biochemical data as an evolutionary clock is to estimate the time of the speciation event. Okay, so it's not to scale here, but they're showing the number of differences in DNA. So we have an early ancestor, we have yeast, very simple yeast, and then we can see the red here are the differences, how far apart the species are. Okay, based on looking at the split in the DNA evidence. So moth, fish, snake, duck, pig, human. So we can see the pig and human are more closely related, and the yeast is most different. Okay, so over time, we can see the differences. All right, so that's where we're going to stop in our notes today. We'll finish this section on cladistics tomorrow. So I don't have a worksheet for you yet. We're not quite far enough to get to the worksheets, but we are going to do a couple exercises from the textbook. I will put this in a homework box below and I'll retype them up as well. You can use either place. We'll do questions 9 through 12. You do not have to do number 10, or I'm sorry, 9 through 12. You don't have to do number 13 using 10 different objects found in your school bag. You don't need to make a dichotomous key. You already did that on Friday. So 9 through 12 is going to be your homework in the discussion box below. Have a good rest of your day.